welcome again. Today we are having our second iteration of Wednesday Wildlife Wanderings. We're happy to have you here on World Otter Day. And today I'm joined by our Interim Zoo Director, David Hamilton. David, welcome.
Hi, happy Otter Day.
There you go. And Dr. Larry Buckley from RIT, welcome. We're glad to have you here today.
Hi, everybody. Happy Otter Day.
So today, David's going to take over our screen, and between the both of them, they're going to give a 20-minute presentation or so. So I'll let David go ahead and start his presentation. We'll take some questions afterwards. So enjoy.
Thank you, Tom. Today we're going to be talking about our genetic project that we started a couple of years ago here at Seneca Park Zoo and Rochester Institute of Technology. So again, I am David Hamilton. I'm the Interim Zoo Director and General Curator at Seneca Park Zoo. And as of today, I'm the Small Carnivore Taxon Advisory Group Chair. So I was elected that, and they let me know today on World Otter Day, so I thought it was very appropriate. One of the jobs that I've had for about 20 years now is the North American River Otter Species Survival Plan Program Leader and Stud Bookkeeper. So I've been keeping track of the otters for about 20 years. And my friend and colleague here, Dr. Larry Buckley. Introduce yourself, Larry.
Yes. Hi, I'm Larry Buckley. I'm an Associate Dean at Rochester Institute of Technology in our College of Science. I'm a biologist, a herpetologist by training. You can see a Galapagos otter in back of me. It's a marine iguana from the Galapagos. And David and I have been working on this project for several years now, and we're really excited to show you some of our results and talk about some of it.
Thanks, Larry. So I'm also an adjunct instructor at RIT, and we've been involved with different classes and different theses programs for different students over the last several years. Today we're going to be talking about our North American river otter. So it's a favorite of lots of people. People seem to really love otters, and especially river otters in our case here. These guys are found all across North America. They were extirpated in some areas like New York State and then reintroduced in many states, including New York State, and Seneca Park Zoo was a part of that program back about 20 plus years ago. The North American river otter is its common name. They are found in rivers, but they're also found in swampy areas, marshy areas, lakes, ponds, coastal areas, in brackish waters. They need fresh water, so they're not like the sea otters where they can stay out in the ocean in the salt water, but they need to get to some fresh water, but they can go out into the ocean a little bit. And so they're mainly right around the shores in those areas. So common names of the North American river otter, the northern river otter, or the Nearctic otter or Nearctic river otter. That just means it's found in the North American zoogeographic region. Their diet is fish and crustaceans, and that kind of changes depending upon the season. They are mainly diurnal, meaning they're active during the day during the winter time, and then they become crepuscular in the summer and when they're around people. So they're trying to avoid people during those times. So they become more active in the early mornings and the evenings. That's what crepuscular means. So there are 13 species of otters found around the world. In Asia, you have the small short-clawed otter, or the small-clawed otter, the hairy-nosed otter, and the smooth-coated otter. Found in Europe all the way to Asia is the Eurasian otter, and they're the one that looks most closely similar to the North American river otter. In Africa, you have the Cape clawless otter, the spot-necked otter, and the Congo clawless otter. In South America, there's the marine otter, the southern river otter, the giant otter, and the neotropical otter, which is in the northern parts of South America and up into Latin America. Pacific sea otter is found around the coast. Used to be found from Mexico all the way up through California, Canada, Alaska, all the way through the islands there and right along the Bering Sea over to Japan. Now it's just found, the southern sea otter is found in California, and then up in Canada, Alaska, and around to the islands just above Japan is where you'd find the northern sea otter. And then our friend that we're talking about today, the North American river otter, is found just about over the whole North American continent, except for the desert Southwest and the extreme cold parts of Canada and Alaska. So I mentioned earlier that I was the stud bookkeeper and SSP coordinator. So my job there is to create a stud book for North American river otters. And a stud book is basically the family tree of all of the otters that have ever been in zoos and aquariums, and where they've been, how long they've lived, all their offspring, and keep track of all those individuals throughout time. And they've been in U.S. zoos since about 1880, starting at the Philadelphia Zoo. And now we have about 274 North American river otters in zoos and aquariums that are a part of the AZA, Association of Zoos and Aquariums. So there's 154 males, 120 females at about 107 institutions. And there are 245 AZA institutions right now. So that's about 43% of all the AZA facilities have North American river otters. And it's about 75% of the AZA facilities that have one of the 4 species that are managed in AZA zoos and aquariums. So the ones that are managed are the North American river otter, the Asian small-clawed otter, the spotted-necked otter, and the giant otter. So the 4 of the 13 species are found in AZA zoos and aquariums. One of the other products that I create with the stud book is the breeding and transfer plan. So I make the recommendations for the different zoos and aquariums to breed their otters, or if they were successful in the past year and have pups, to move them to a new facility, or to if a new place is coming on board, then help them find their otters from one of the other facilities. So I make these recommendations so that we try to keep up the population. With these guys, we also have some coming in from rehab facilities. So since they're native here in North America, there is some chance of some that are orphaned or are found and then raised. They aren't able to be released for one reason or another, and then they might be placed within a zoo or aquarium. So that's another place where they sometimes come if we don't breed them within zoos and aquariums. We are wanting to boost our success rate with them, and that was some of the problems that we started seeing, and why we started this project that we're talking about today. So for creating the stud book and the breeding and transfer plans, there's lots of different software programs that go into doing that. There's PopLink, and now ZIMS, Zoological Information Management System, has a stud book feature now. So we're able to keep track of all of the otters online and connect to all the other zoos and aquariums data live. So as things are updated, if an animal unfortunately dies, or they have birth, or some other change in the population or with their animals, then we're able to get that updated pretty much instantaneously now. Before, I'd have to ask for updates every year and then put lots of information into the software database and do lots of typing there. To create the breeding and transfer plan, there's also a specialist that helps me, a population biologist that works with the software all the time and helps create the breeding and transfer plan. She has lots of experience with lots of other species and knows the software in and out, so she's able to help me make those recommendations. So after requesting all the information from the different facilities, I compile all that, bring it together and say, okay, this zoo wants to get a new otter, this zoo needs to send out its pups, this zoo wants to breed, this zoo doesn't want to breed anymore, and I take all those wants and needs and create the breeding and transfer plan, put out those recommendations. So in that, then we started seeing that we weren't having enough breeding. So there was some question as to why. So there's a few different master's theses at RIT that kind of looked at that, and we were seeing that there was something there, some kind of issue out there why we weren't having a lot of success. So one of the things that we were considering was that it might be a subspecies problem. Or it could be some kind of latitude difference. So they might be an animal from Florida, and we're trying to breed with an animal from New York, and they never have any reproductive success. Maybe there's some issue there with their windows of fertility. So maybe the male is fertile and goes through his cycle before the female even comes into estrus for the year. So we were looking at all kinds of breeding issues like that. It seemed that some of the zoos, especially those kind of mid-latitude, middle of the country, were the ones having more success. So a few years back, I made some recommendations to kind of switch animals up and get the northern girls with the northern guys and the southern girls with the southern guys, and try to get them, not move them too far in difference in latitude, and try to have some success. And we did there. So I wasn't sure whether that was because of the latitude change or because of just a change in their life that kind of shook things up and created some success back then. Formerly, there were about 20 subspecies of North American river otter, but it has been kind of whittled down to about 7 subspecies. And 3 of those are more common within zoos and aquariums. So the Lontra canadensis, Lontra canadensis lataxina, and then just a few Lontra canadensis pacifica, those from the West Coast. So we were kind of looking at those to see if there was any kind of issue there, anything that we can kind of track, and that's kind of what spurred the genetic project that we have going now. Just for some more background, North American river otters belong to the Mustelids. That includes the badgers, the weasels, the otters. They are from the least weasel, which is less than a foot long, all the way up to the giant otter, which is almost 6 feet long. If you remember way back from science, you have kingdom, phylum, class, order, genus, family, genus, species. So here you have the Animalia kingdom, the phylum Chordata, subphylum Vertebrata, meaning it has a backbone. The Mammalia class, they belong to Carnivora, meaning they are all in there with all the cats, the dogs, the bears, all those are a part of the Carnivora. Suborder Caniforma, meaning it's kind of dog-like. They have a muzzle. They are small plantigrade animals. And family Mustelidae. So I am going to pass it off here to Dr. Buckley.
Great. Well, thanks, David. And again, welcome everyone. And just to continue on to give you some of the background for the study that David and I are working on now, appears to be the case. The family that were in the Mustelids are probably a Miocene or origin group. I believe that some of them were a little older, some of the fossils, but so we're looking 20, 30 million years. But actually, the species we're working on, the North American river otter, is much more recent. And so what few remains there are that predate humans don't appear to exceed 1 million years. So we're dealing with a much more recent organism with the North American river otters. And because of that, the next slide shows you exactly what that meant and has meant, or what we think it has meant for these otters, which is that the global ice cap over the North American continent has been probably quite a big driver in the evolution of this species, just because it has both facilitated where it can go, and also at times in the last 2 to 3 million years, while that ice sheet's been there, it's probably blocked a lot of where that species has been able to distribute itself. And so we were wondering when we started this survey both about the reproductive consequences of the wide distribution of these animals from Alaska all the way to Florida and everywhere in between, or almost everywhere in between, and also just its history over the last million years or so. Did it recently get to those areas that were formerly glaciated, or has it been there since the glaciers left, 10,000, 20,000 years ago? Is it a result of inland distribution, migration, these animals going over long periods inside the country, in the interior of the country, or have they migrated along the coasts, right? And all these kinds of questions were things that we were interested in finding out. The genetics themselves started with some samples that we got from blood samples from zoos. David has been great with all his connections at being able to convince zoos that normally do blood work every year on these animals. They check them for parasites and just general health. And so zoos will generally take blood samples. And so zoos have been very generous with us. David's been able to get us over 100 samples of otters from different zoos. And we use that blood to get DNA out of. The next couple of pictures are us collecting some samples. This is myself collecting a sample from at the State University of New York. The New York State Department of Environmental Conservation has a program in which animals every year that are bycatch, that are caught accidentally or road killed, are turned into the state, and then the state allows biologists and others to have access to those for in our case, for research purposes. So we were taking tissue samples there. We then get those tissues, or in the case of zoos, their blood work, blood is shipped to us. There's David in our lab holding a tube of blood from a zoo, and we extract the DNA out of those tissues, whether it's muscle tissue or what more commonly for us, it's been blood. And we extract that DNA out of there, and the reason we need to do that, of course, is when we want to sequence that DNA in order to compare it among samples, we have to have that DNA in a very purified condition. So we have to treat it with digestive enzymes and then clarifying chemicals that both extract it away from the other cellular debris that it's mixed in with, but also protect it long term. So now what we have in our freezers are permanent genomic stocks of the genomes of all of these otters that are in our study, and we can then dip into those stocks at any point, pull out a little tiny amount of DNA, and go ahead and do the sequencing on that DNA to find out what those sequences are. And we're going to show you a few of those here in just a minute. To place the genetic project in context, what we did was we took the tree of the entire family Mustelidae, or at least the latest tree, and what these trees represent are relationships among living species, among the species that have been sequenced. This is a DNA sequence tree resulting from DNA sequencing. And so you'll see otters there at the top. Just to put this in perspective for you, can you look at the top? See the yellow part? That's Lontra canadensis. That's the one we're working on. Look at all the other Lontra species, and Lutra and Mustela and Martes and all the other genera of Mustelids. You can see from the tree that we're showing you that the Lontra group that we're working on, Lontra canadensis especially, is a relatively recent development within the family, the Mustelid family. And so it's consistent, the DNA data of all Mustelids is consistent with what we're seeing from the geological record, which is that Pleistocene glaciation has pretty much affected, and probably within the last million years, isolated the northern otters, the canadensis otter that we're working on. So that's kind of good to put that in perspective and see where we are in terms of the family. The next thing we did was we sequenced the mitochondrial, a portion of the mitochondrial genome, a gene called cytochrome B, which is a really great gene to work with, from all of the otters so far that we've been able to acquire. And if you look closely at this tree, what you're going to see is a number, and then after each number, like number 108 there, you'll see two letters that correspond to the state that the sample was acquired from. So there's that very first one, there says 108 New York. That's our 108th otter, and we got that from a sample in New York, and it actually had locality data associated with it. So what we have associated with all of these otters, and each one is, like I said, a 2 or 3 digit number and then a 2-letter state identifier in back of it. Many of the otters are from zoos that we know the state it was from, but we don't know exact locality data. Other otters, for instance, particularly the ones from New York, we have very precise locality data. We sometimes know the exact latitudes and longitudes that those otters were acquired, those samples were acquired from. In other cases, we only know the state that they were acquired from. And the second thing you're going to notice is that notice how all of the lines on this tree are very, very close to one another. They're very short. These otters are very closely related to one another. And then at the very bottom of the tree, we call that the root of the tree, the really long line that leads down to the first two at the bottom, which is L. felina and L. longicauda. Those are two outgroup otters. Those are two otters that aren't the North American river otter, and we use those to root our tree. And it looks like a root there, and you can see that on the left and the bottom left corner. You see how much further the line is leading to them. That means genetically they're much more different from our otter than any of our otters are from each other. So the North American river otter looks like a good species. There it is. They're relatively closely related. There's one interesting group that we'll show you on this. Let's go to the next slide of the next tree, and I think this tree is a little bit easier to see. There's one group, I've taken out the outgroups on this tree so that these are all North American river otters now. None of these otters are different species. These are all our North American river otters. What you can see is there's a deep split at the base of the tree here, and that splits most of the otters above and below that point. And below that point, you'll see otter number 3 from Florida, number 5 from Georgia, number 17 from Florida, number 68 from South Carolina, number 106 from New York. That group is distinctly different from the remaining otters that we've sampled. As a matter of fact, some of those samples are otters that are genetically identical, and the ones that are genetically identical on this tree are right next to one another. So if they're printed on the horizontal right next to one another, it means they have the exact same genetic code for the gene that we sequenced. And if they're on different branches, they have a different code. And so what you see is that the first thing that we noticed was that there's a split between deep in the tree, in the North American river otter tree, between some organisms that appear to be on the East Coast, particularly in the Southeast United States, and then the entire remainder of the distribution. And by the way, I might remind you, even if you look up to number 19 and 20, which is sort of buried in the middle of the tree, just underneath that very long line of letters and numbers, there's ones that say AK, which stands for Alaska, and then WA or OR, which is Washington and Oregon. We thought those might be very different from all other North American river otters, and turns out they're not all that different. They're much more similar. The really different ones are in the Southeast coast, and that's something that both surprised us, and also we're still trying to figure out the exact meaning of. But the data don't lie. There they are, and you can see that there is a distinct difference there. The next thing I'd like to show you is, instead of a tree, it's what's called a network analysis. Oh no, this isn't. This is still an unrooted tree, and I'll show you the network analysis in just a second. This is a little different version of the tree. I told the software to put the tree in a little circle so you could much more easily see the groups, the closely related groups of otters. So off to the far right here at about 3 o'clock, if this were a clock face, at about 3 o'clock, notice the Florida, Georgia, and New York group, that group off to the left here as distinctly different from the other otters that are in here. The next slide has the actual raw data, an example that, and David and I wanted to show you this because we wanted you to see what kinds of things we look at a lot of A's, C's, T's, and G's in the lab when we get this, when we sequence these otters and get the data back. It comes as a series of the, remember the 4 letters, right? A, C, T, and G, the nucleotides that make up the DNA molecule. And what you'll notice if you look along each one of these otters, right, is that every once in a while, there'll be in the vertical, like at base pair number 852 there, which is about in the middle of the screen, everybody has a T except for 3 otters that have a C at the same position. So the key here is to go from the top to the bottom and then compare the otters and see which ones possess the same nucleotide at the same position versus those that have a different nucleotide at the exact same position. And otters that have different base pairs at the exact same position as other otters have what we call a single nucleotide polymorphism at that place. So that's a distinct difference that they share with other otters that have that same difference from otters that have a different base at that same location. The 2 otters at the very bottom of this table are the 2 outgroups, Lontra felina and Lontra longicauda. And those are the very 2 at the bottom. And notice their nucleotides, we left them as small case letters so we could tell them apart more easily. Notice how they have a lot of differences from our North American river otters. All the rest of them in that list are North American river otters. And you can see from this, we call this an alignment, kind of for obvious reasons because the genes are all lined up with each other. You'll see most of the North American river otters are very similar to one another. Most of those bases are identical, and maybe there's just a few of them that have differences. Okay, so that kind of gives you an idea of what we mean when they say they're genetically different. What exactly do we mean? And that's it right there. Those are the differences. So the next graph I want to show you is a graph that is called a network analysis. And the network analysis is even more interesting than the tree because not only does it allow me to show you the differences between the otters on the ends of branches. So notice the top otters, the top 3. There's number 3 from Florida, number 68 from South Carolina, and number 106 from New York. Notice how those 3 are separate from all the rest of the otters. Those are those 3, and actually number 3 Florida is the same as 2 other otters. So not all 5 otters are listed there because it lists otters with identical genotypes in one circle. You don't get, each otter doesn't get its own circle. Every one of these circles is an otter that had at least 1 nucleotide difference from every other otter. And if they were the same as every other otter, they're just included in the same circle as the otter they were identical to. So that's the first thing. The second thing you're going to notice is, notice how there's a little number along each of these lines. That's actually the number back in that previous graph with the DNA data that corresponds to the actual nucleotide difference that that otter has with another otter. Notice that Florida, South Carolina group has many more differences along the line leading to it. It has 1, 2, 3, 4, 5, 6, 7, 8, 9, sometimes up to 12 differences along the gene we looked at relative to the other otters in the list. So it has a distinctly higher genetic distance from the other North American river otters than those otters do from one another. The typical distance in the other otters is maybe 1, 2, or 3 base pair differences from one another, whereas those Southeast U.S. samples appear to have upwards of 8 to 12 differences from the rest of the otters. And so I like this network version because it's easier to interpret. The second thing that it does that makes it better than a tree for looking at relationships within a species is that it shows you which otter haplotypes, and when I mean haplotype, I mean one of those gene sequences that we just looked at, which ones are ancestral to others. Because all of these otters are still alive, it means that some of those sequences actually are the ancestral sequences to other living otter sequences. So some of the genotypes that the otters now have are actually the ancestors of genotypes that are still alive, and the ancestors are still in the population. And because mitochondrial DNA evolves so rapidly, you do have existing sequences that are the ancestors to other sequences, and the ancestors and the descendants are still all alive, and those gene sequences are still present in the population. a tree doesn't really show it to you that way. So this is just, I think, a clearer way of seeing the differences between them. David, did you want to add anything? I kind of went quick through the trees and things. I didn't know if you felt like adding anything in there before we take questions from folks.
No, that's great, Larry. Now I'll be able to take some of this data and try to compare up different pairs to see if we've got the right ones, to see if they're breeding or not breeding. And then maybe the genetics is part of it. Maybe there's some other reason as well. But I'll be able to use this data along with all the stud bug data to now try to be able to have more success with breeding the zoo population of otters.
There's a good question, David, in the chat room, that is, if the location is a zoo and the zoo's location is used, what is the origin of the animal originally and how we knew that? That's a great question. And David and I can answer that a little bit. David, I might let you do that because you know more about the localities at the zoos than anybody.
So I have all the records from the zoos and aquariums, so I know where they think it came from. I'm not just saying it was a zoo in Tennessee. I'm saying where that zoo thinks it came from. So if they know that the otter came from Kentucky, then I will put that in. I don't put the institution's, the zoo's address in as my location.
Do you have those locations for pretty much all of the samples that you have, or are there some dead ends where you don't?
There are some in here. Like right about in the middle, you'll see number 23 is unknown UN. So I don't know where that one came from.
With enough data, hopefully at some point you should be able to pinpoint that, correct? With enough samples?
If we have enough, yes.
Yeah, and the great thing as well is with these haplotype analyses is that David and I, once we get enough known samples from known localities, we can also help reverse engineer where we think a sample probably originally came from, even if it's unknown originally, even if it's unknown by the time we get it.
Right, and one of the confounding factors is states have reintroduced otters and they pulled those from other places. So we, even though if it is from Pennsylvania, then its ancestor may have come from Louisiana originally.
Yeah, and that's shown real clearly. Can everyone see? Can you look at this network closely? You'll notice that New York is represented in two places on this graph. It's represented in the one at the very bottom, the group at the very bottom of this graph. See 117, 108, 109. It's also represented, oddly enough, way up in the top right corner near that unique southeastern United States group. It's also, there's a New York animal that we sampled that has the genotype that belongs in with those southeast United States animals. And I believe, if I'm not mistaken, David, there was also a New York one, but I think he's buried because he's identical to one of these others that was near the Pennsylvania groups, but I don't see him anymore because this is a condensed version of our full network.
Yeah, I think that was...
So New York is actually located in 3 places on this network, depending on depending on where the otters, when the otters repopulated the state of New York after the glacier left, whether they probably populated it from the East Coast, populated the state from the central part of the country, or were very recent additions to the state, perhaps from introduced populations that were reintroduced into Pennsylvania and might have come northward naturally in the last probably, I don't know, David, 20 or 30 years maybe.
25, 30 years, right.
Yeah.
We also have a couple of individuals that we know came from Alaska, and we're able to compare them and how far apart they are from those in Florida and everywhere else.
They're in the middle there, number 19 AK. I believe the 2 Alaska samples have the same genotype, so they're both included and only 19 shows up there. Right, and interestingly, right, it makes sense. They're most closely related to what? Number 25 from Washington. So the biogeography of the otters does make sense. That the genetics and the biogeography appear to agree with one another in the sense that we're finding a lot of otters that have similar genotypes from areas that are roughly equivalent or close to one another. Florida and New York are examples of otters that appear to have genotypes from many different places. And that's an interesting question that we're looking at with more data, hopefully.
Great. Thank you. David, there is I think you answered this at least partially earlier on about the giant otter. Can you just speak briefly about the difference in the size from the North American river otter and giant otter, just to give people some perspective?
The giant otter is almost 6 feet long, whereas the North American river otter is maybe 3.5, 4 feet long. So giant otter, if it stood up on its hind legs, would be almost as tall as I am.
Yeah, great. So that's all we have for questions. Dr. Buckley, I want to thank you. David, you've worked at RIT for quite some time now, so I know that you've been integral in keeping our partnership going. And this is a great representation of how important partnerships are between academia and zoos, and how we can really leverage each other's what we do well and help with things that maybe we don't do well, like genetics. You know, zoos don't really do that a whole lot. So I want to thank you both for coming in, and we'll see you guys later. Thank you very much.
All right. Thanks, everyone.
Tom, happy Otter Day, everybody.